Yeah, so today, basically, I'm going to just be covering some new features in the spatialLIBD package um, that related to plotting specifically. Um, so yeah, I guess I'll just dive right in. So one of the big functions in the spatialLIBD package is sort of plotting spatial data, just, Visium data in particular. And so there's these functions visgene and visclass. I'm not sure how many people are familiar, but the idea behind visgene is you're just plotting just one of the continuous variables that spatially for one of your samples. So in this example, in this vignette, oh, by the way, this vignette is on the RStats sheet if anyone wants to follow along. So in this case, if we just load some data that's provided by the spatialLIBD package, a small subset of the spatial DLBFC data, basic usage of this gene. I'm going to skip over just a bit. Just looks like this. So you give it your spatial experiment object, you give it one gene, and then it just plots it spatially. So that's the idea behind visgene. That's how it's worked for a long time for spatialLIBD. So you may be familiar with that already. In this vignette, I'm going to do some more complicated stuff. But we start by just loading some white matter genes from the literature. So we just have 4 of them right here. So in that example, we just plotted the first one, GFAP. So yeah, very typical usage of visgene here. So in terms of the biology here, so GFAP normally uses like I said, a marker for white matter, but also it does have good expression in layer 1. So in this tissue in particular, it's a little bit small here, but yeah, this band mostly, the little V shape is mostly layer 1. So this gene in particular is not super selective. So this piece of the vignette actually goes more over that. So another thing that's provided by the spatialLIBD package is the modeling results for this data. So something you can check actually is you can see so how this gene ranks in terms of the modeling results. So if we actually check the rank here for GFAP in terms of in terms of white matter, it's actually ranked 386th. So it's not super. It is a popular marker, but it's not super high. So yeah, here's actually the little plot of how it is distributed across the layers. So yes, it's definitely highest in white matter, but we get considerable expression in layer 1 too. Layer 1 also. So I guess this is sort of related to what Louise showed in her talk, where if you do a 1 versus all strategy, one problem with that is sometimes the second highest expression is pretty considerable. So you wouldn't see that in the mean ratio method, for example. But nevertheless, it is a popular marker, and it does do well on the 1 versus all test. But this is sort of another example where maybe sometimes it's good to check in a sort of a data-driven way that how your marker performs. Because sometimes you can find markers in the literature, but they're not necessarily the best for your particular data. So yeah, so this is the normal way you use visgene, just one continuous feature. So to actually get to the most recent updates, so what we added that's important is now you can provide visgene a vector of genes or a vector of continuous variables. So you might wonder how that works. The idea is that you're only plotting one. Plots will look similar. You're only plotting one variable. It's just the variable is combined across the continuous features. So I'll describe how that works. We provide 3 different methods in this package for combining. Obviously, there's a lot of ways that you could potentially combine continuous variables into one visual illustration. So the default method that we provide, and by the way, actually, I should mention that the parameter name that we added to do this is called multi-gene method. So that's controlling how the continuous features are combined when you do provide more than one. And the default is it's called z-score. So just to go into like the idea behind the math of this, yeah, I guess I'll describe the three methods and then you can sort of make your own opinion about which one you think is the most like theoretically reasonable. But in terms of the z-score, the idea is that like, so now we're going to plot, remember we had 4 genes here in this vector. And then we have just one plot. The idea behind z-score is that you're taking, I guess, sort of the most naive way to combine expression, if you're just thinking about how to do it without thinking deeply about it, I guess, is you might just average the expression and show that. The problem with that is that you would then have genes that have like the absolute expression and the absolute variation would matter. So you get like genes that with higher expression dominating the plots, which isn't necessarily what you're probably looking for. So the z-score method sort of normalizes the expression of each gene individually to be a z-score. So you consider all spots in the tissue, and then if a particular spot for one gene has a z-score of 1, that would mean like its standard deviation is it's one. The expression is 1 standard deviation above the mean across all the spots for that one sample. So each gene gets a z-score for the spot, and then to display it, we just average the z-scores. So that's sort of a way to make the methods insensitive to like the absolute expression levels, and you don't have genes that vary more dominating the plots. So you sort of weight each gene equally in terms of the variance. But in terms of visually what that looks like, I guess I'll compare this with the plot buff, but we do see, now that we're using more white matter genes, we do see more expression in the white matter area in this bottom right corner. We still do see like layer 1, probably because we only have 4 genes and then one of them is definitely also a layer 1 marker in some sense. But if we go compare it to before, this one, was layer 1 dominating here. White matter was just sort of like almost part of the background. Yeah, so that's the first method. That's the default. If you just do VisGene and you give it a vector, that's what it's going to internally do.
How many genes can you send in?
As many as you want. So it could be, yeah. In theory, I guess you could do every single gene if you really wanted to, but
I mean in the vector, in the additional vector, so you can send.
Yeah, as long as it's in the data, it'll do it. So yeah.
What if it's missing, like you don't have any gene expression for a point?
So if it's like NA, it does just drop NAs.
So you just average the other 3.
Yeah, it would just ignore any NAs as long as there's not like, yeah. So it's just a couple, it'll be fine. Yeah, in terms of the other 2 methods, the next one's kind of harder to describe, but it's called PCA. The idea is that you, basically you take a matrix. So in this matrix, you're going to have the continuous features as the columns of the matrix, and then the spots would be the rows, and you're performing PCA on that, and then plotting the first principal component. So like the idea behind that is that, so the first PC is going to point in the direction of the maximal variance. I guess in English, it would be sort of like you're capturing like the dominant spatial signature of the feature set. So one thing that makes that a little bit more complicated is that like the sort of the direction of principal components is arbitrary. In terms of like, not arbitrary, but like you can flip it. I guess, okay, describe this better. Like, since you might get a plot that looks like this, but exactly inverted if you plotted the principal component, because if you negate the principal component, it still points in the the direction, like this along the axis of highest variation. So that component of PCA is sort of arbitrary. So there's a little trick that we do internally is that we take a look at the rotation matrix and basically if the coefficients in that from the rotation matrix for the features are mostly negative, we multiply the principal component by -1. So that's like a little trick that we do to guarantee that like if you use the inputs, if the inputs tend to have higher expression in one area, then the principal component is going to have higher value in those areas as well. So because otherwise, like I said, you might get a plot that looks like the exact inverse of this, and then that's not really what you want to see. So this method I think works. I mean, when I've experimented with it, it tends to give like the cleanest results of the three methods. So yeah, we can compare it maybe against, like here we see the obviously great signal in the white matter region. Pretty similar to the z-score. If I go back up, it's like, yeah, fairly similar, maybe slightly cleaner. But obviously, yeah, it's like data dependent. You just have to try this out with your own data. Yeah, the third.
Wait, Nick, I
final
have a
method.
question about the PCA.
Yeah.
Are you able to access like the, I guess like the PCA calculated like overall for the data like that lives in like the reduced dimension slot of the object?
Oh,
yeah.
So like, are you saying like, do we use actually, sorry, I don't know if I understood.
Yeah, so like in the SP object, there's like the reduced dimension slot, right? Where there's like
Yeah.
the PCA for like the overall data set.
Mhm.
Are we able to access that with any of the viz functions yet? Or like, I forget if
No, I mean, not currently.
Okay.
Not sure if that, I'd have to think about how that would work, but, yeah, I guess that's a good idea, because it would be like spot level data as well, right?
Yeah, that could be neat. I get that it's like it's different from what you're trying to do here, but got me thinking.
No, yeah, that's a good idea, actually. We might be able to incorporate that. Yeah. Because it is one data point per spot, and then
Mhm.
yeah, that's cool. That's a good idea.
All right, cool. Thanks,
Yeah.
Nick.
No problem. Yeah, so the final method, yeah, ignore this really ugly plot for now. The final method is called sparsity, and I'll explain why it doesn't work very well for this example. But the way it works is that you take basically, it's somewhat simple to describe. You just take the proportion of features that have positive expression for each spot. So we've seen this work pretty well in other cases. In this case, we have 4 features, right? So that means that the plot here can only hold values of 0, 0.25, 0.5, 0.75, and 1. So like, even though there's a continuous scale, there's really only like 5 possible values, and then that's not very informative in this case. So this would be a method that's more appropriate when you have more features. So that way you get more continuous values. So we wanted to show it here, and then even though we have 4 features, like this is sort of a caveat, like make sure if you use this method, just probably make sure you have more features. So
This is just a binary, like the genes in the cell or in the spot or not.
yeah, and it's doing it, yeah, and then there's 4 of them. So yeah, so there's only like a few increments that are possible. Yeah. Okay, so that was an example of 4 white matter genes. To give you ideas of what else you could consider doing using these this new functionality with is so instead of just using 4, you could actually find marker genes in a data-driven way. So from this data, if we just take like the top 25 or 50 white matter genes, what does that look like? See if we get better signal with this. So if you just use 25, this is what it looks like. So not super, pretty noisy. And then we can sort of compare the different methods. So this is z-score, top 25. And then top 50 actually, in this case, looks like we're getting better signal here, right, compared to this, just a little bit. So we did it with all 3 methods and just check that out. So PCA, a little bit less noisy even at 25. At 50, it's looking pretty good. Other than I mean, we still do get that band of like layer 1-ish spots. And then I said it would work better for more markers, but yeah, very noisy. I mean, we do see more expression here, but you know, there's something that maybe I might implement is like, I think you'd see better signal if you chose the cutoff to be different than 0 in this case. So actually, no, this is already implemented. I was thinking something different. So there's another parameter in VisGene called min count. That's defaults to 0. So if you had chose the threshold to be like, I don't know, 0.2 or 0.3 here, maybe you'd see these spots drop off. And then, so there's some visual stuff you can do to make this a little bit cleaner. It's an option here. So that's 25 and then 50. In general, we're seeing more marker genes make the signal cleaner, which is not surprising. But yeah, we can play around with the cutoff maybe here and see if that cleans things up as well. So yeah, these are examples with genes. I did mention I was using the word continuous feature for a while, and that's because you can also use variables that are in the call data. So any spot level data, really. I guess not the PCs or not reduced DIMs, but anything in the call data. So another example is like if you just, in this example, we're just plotting like some gene and some UMI, so it's just like quality metrics that are stored in the call data. So you can really combine whatever you want. Also, I guess I'll show this later probably, but you can actually mix and match. So in theory, if you wanted, you could put genes in and call data variables, and then it'll combine in the same way it works for everything else. You can get, I guess, get creative with that. There's lots of things you could do.
And it's the average for its plotting? The average of the combination of the, you know, if I have to continue variables there.
Yeah.
It's averaging each one in plus 1.
Yeah, well, it's taking, in this case, it's taking the z-score of each across all spots and then averaging those z-scores.
Okay.
That's for this, yeah.
Mm-hmm. Yeah, you're right. Z-score. Thank you.
So another thing we explored here in this vignette is, so we also, in the DLPFC data, we also have like spot deconvolution results. So I presented about that. They were pretty noisy, but in this case, there's actually, interestingly enough, that we can actually use this to clean up some of the plots from earlier, and I'll show you what I mean in a second. If we just plot the, so in this data, we had 3 different software tools that we used to perform spot deconvolution, and we also had 2 different cell type resolutions. In this case, at the broad cell type resolution and using the software Tangram to predict the astrocyte counts. That's what we're exploring here. Obviously very, like I said, like these results were pretty noisy. We're not getting anything informative in this plot here, but interestingly enough, if we combine that with GFAP, then you get a very clean plot, which is kind of a little bit surprising because if we, okay, if you just remember this plot, I'm gonna go back up to the GFAP one. It's pretty high up here, so looks like this, right? So the idea here is that like astrocytes are going to be sort of co-located with GFAP and sort of they're sort of should be similar signal. And it just in this case, it seems to clean up noise using the PCA method here. So that's just like an interesting finding where you can consider combining spot deconvolution results with genes and get clean signal. There's a lot more you could do here, but this is like a start with the, oh my gosh, I forgot to preload this with yeah, a lot more you could do here. So this is just using the VisGene function from the package, but I'll also show that like with this latest version, we also updated how Shiny apps that use the latest spatialLIBD work. So these features are implemented in Shiny apps. These take a while to load, and I had forgotten to load them, but we're gonna in a second we'll explore a couple of them, show what that looks like.
Certainly, just to be sure, if I understand, when you are using the method that use the PCAs, you pre-calculate the PCAs or the function is calculated?
The function is actually calculating it. Yeah, it's not using anything pre-calculated.
Okay, igual I understand. Just I want to be sure. Okay.
And it's only doing it for the one sample. There's another detail that, like in theory, you could compute it for all samples and then just plot one, but this plot computes it for one. So, yeah. Okay, so the spatial DLPFC Shiny app we have, and actually we did update it with these new features. So just to show what that looks like. So if we go to like spot level data, which is what we've been exploring. So I guess what we did in the vignette was sort of like the gene static. They're just displaying genes. So yeah, by default, the app will show like we have this sample, and then we have we go down to the continuous variables to plot. We just have some UMI right now, which is how it, again, just one variable. So what changed here is basically now you can do like if we could select this as well, and then we have two things selected. And then the default method is z-score, like I said. So that's also a new tab here, multi-gene method, right? So I described 3 methods. So that's available on the tab right here. Something that might be surprising if you don't, if you haven't used this is like, I'll just do an example where, okay, we were talking about GFAP. I select that. I mean, obviously this is sort of like a random choice of 3 variables, but just to demonstrate like something that we may change maybe. Something that makes it tricky is that like you have to sort of remember which things you had selected. So like GFAP is way down on this list. If we wanted to unselect it, we have to search for it and then unclick it. So that's sort of like something that's not the cleanest right now, but so I guess just a caution to remember things you have selected, unless you want to reload, which takes a while. But something else we can explore is so like I said, we have spot deconvolution results from 3 different software tools. So one thing we could check is we can sort of form like almost a consensus of results among the 3 tools. So in this example, I'll just do like excitatory layer 5 cell type counts as predicted by each software tool. So we have cell to location has one tool, spotlight I'll select as well. And then 10 gram. So actually, do I still have, it's okay. The reason this isn't clean is because I forgot to unselect some UMI, I was wondering. It's still a bit noisy here. I bet you if I unselect spotlight, which was our worst performing method, you might get, okay. So that's a little bit clean. We could also add in, there's all sorts of things you can do here. You can add in TCP4, I believe, is a marker for layer 5. Yeah, so just to get your creative juices flowing, right? mix and match spot deconvolution results with genes that are markers for specific regions of the TLPFC or whatever tissue you're working with. So that's something that can be done here. So we also implemented things like the other tabs work as well. So like there's the gene interactive tab. So once that loads, you can do anything you could have previously done with the gene interactive tab. So you can select a specific set of spots. You can label them as like, I don't have a good label for this. Actually, you know what? Maybe we could do something better. Let me unselect this. Actually, I don't remember how to unselect. Okay, we're just gonna go with this. So we can label them and go to the clusters interactive tab. And then go to the manual annotation that we just made. I believe, okay. Yeah, sometimes it says that before it loads. Let's give it a second. But yeah, you have like the plots, the continuous plots measuring the multiple variables at once, and then spots that you labeled, and then showing where they lie in lower dimensional space. UMAP space. But you know, all the features of spatialLIBD app work with this new feature now. So let's see. So this, yeah, not all the apps have been updated yet with the latest spatialLIBD, but if you make your own and use the latest, then you'll get these features. Just another example is we have the spatial NAC project, also has this working. So this was an interesting project because we had to stitch together multiple capture areas computationally. But yeah, just another example of like we wanted to let's see what pick GFP, and then we'll pick MOPP as well. And then, yeah, so 2 white matter markers, and then you see it, you can explore how it works in this data where we have the multiple capture areas and it goes smoothly across the capture areas, and that's a lot to explore here. So I'm not sure of the full list of Shiny apps that are working right now, but I know we're actively getting those updated for the ones that we have previously published and stuff. Yeah, I think that's all I had for today. So
Oh, it's okay, Nick. I know that I can plot, for example, the white matter, because I want to make a test to see if what I see in the image is white matter now. I
yeah,
know that I can send any variable that has continued trend.
yeah, it's like
So
good to combine,
yeah.
like, because each gene can be like noisy by itself, and yeah, you can get sort of consensus with this.
Nice.
Cool. Yeah, happy Friday, everybody. See you guys next week.
Happy Friday.
Great work. Thanks, Nick. Those great updates.
Thanks.